Let's begin our discussion of polysaccharides now. We sort of graduated from disaccharides. We talked about lactose and sucrose, whether one is reducing versus one is non-reducing. And extending on that, we're going to go into multiple monomers, hundreds, thousands, even tens of thousands of individual sugars linked together via glycosidic bonds. So we have to remember that glycosidic bonds are named by the anomer that is forming the bond. If it's an alpha anomer or a beta anomer, we have to designate that in our glycosidic bond. So our first example here is a polymer of glucose known as cellulose. So cellulose is glucose held together via a beta-1,4 glycosidic bond. So it's very important that we know it's beta-1,4 because remember, the anomeric carbon in aldoses such as glucose is carbon 1. So that's actually forming a bond, and in cellulose specifically it's the beta anomer, and it's forming a bond with a number 4 carbon of another glucose. So this number 4 carbon is not the anomeric carbon, so it's just beta-1,4. It's not going to be anything else. So it's a beta-1,4 bond, and this is linked 15 to 20,000 or so times. So you have basically a polymer of glucose, beta-1,4 linkage. We see this a lot in plant cells. We see this a lot in bark of trees. This beta-1,4 linkage has many types of properties. One of it is that it actually forms a very fibrous material, kind of like a rope, so it's very tough. And humans don't have the enzyme to actually digest this, but a lot of other organisms do, particularly elephants or gorillas, baboons. So these other types of mammals, these other types of vertebrates, not humans, can actually break this cellulose with digestive enzymes and liberate free glucose. But we do not have the bond, the enzyme, to actually break apart the beta-1,4 glycosidic bond. So remember, this is a polymer, so many, many thousands and thousands of units. They arrange themselves in sheet-like structure, so it's a big sheet. One thing you want to notice here, one sheet here, here are your glucose polymers, and then the other sheet is sort of flipped. So one residue is flipped relative to the other, one residue is flipped relative to the other. So you have planes and planes and planes that are linked together via these hydrogen bonds, shown as blue dots. Also, the planes are staggered, so it's not stacking, but the stacking is sort of shifted relative to the other sheet. So that molecular arrangement gives rise to something that's very tough and fibrous, almost like a rope, very hard to digest. We do not have the enzymes or the proteins capable of breaking these bonds. All right, so a polymer of glucose linked together via a beta-1,4 glycosidic bond is cellulose. We're going to flip that and make the glycosidic bond alpha-1,4. So the alpha-1,4 glycosidic bond comes in very different reincarnations. So one reincarnation is where you have a short amount of glucose monomers, so we're going to call that amylose. Amylose is basically not a lot, maybe several thousand or so units of glucose. So the length of the polymer distinguishes amylose with amylopectin, which in turn discriminates the big one, which is glycogen, which is a fundamental part of a liver. Okay, that's your glucose reservoir for the liver. So we have an alpha-1,4 polymer, a beta-1,4 polymer, as alluded to before, cellulose. We cannot digest this, but humans definitely have the capability to digest the alpha-1,4 glycosidic bond. Another thing that we want to discriminate between these lengths of these alpha-1,4 polymers is branching. So a lot of alpha-1,4 linear linkages are branched together with an alpha-1,6 shown here. The alpha-1,6 branching occurs most extensively and to a great extent in glycogen. So the beta-1,4 bond is sheets and fibers, staggered, very much stabilized by planar hydrogen bonds. The alpha-1,4 polymer is a helical coil, okay, not an alpha helix. Okay, so Don't think this is an alpha helix. This is not amino acids we're talking about. We're talking about sugars here. A polymer of glucose linked together with an alpha 1,4 glycosidic bond has this helical coil, again, also stabilized by hydrogen bonds. So fundamental differences in the structure. Amylopectin has alpha 1,6 branch points. It's the same alpha 1,4 glycosidic bond units, but we do have branching. Not as extensive as glycogen, but we do have branching. The difference between amylose and amylopectin is that one has alpha 1,6 glycosidic bonds. Amylose is generally considered starch. Amylopectin is found in a lot of fruits and starchy vegetables, mostly potatoes. Okay, now let's go to the big one. So the big one here is glycogen. Very fundamental aspect of your liver. You can actually see glycogen granules in liver cells under an electron microscope. And we're talking about very extensive alpha 1,4 polymers and also very extensive alpha 1,6 glycosidic branches. So glycogen is its own science. It's its own subject matter. We're only dedicating 1 or 2 slides to this. The alpha 1,6 branching is exquisite. The regulation of glycogen is extensive because you see in the liver that's your storage of glucose. So if you're exercising, the body needs to have access, specifically the liver, to take glucose from the glycogen polymer. Or if you're in a resting state, if you're sort of sedentary and not in a great deal of physiological threat, the reservoir of glucose can be incorporated into glycogen. So we have glycogen breakdown and then glycogen synthesis. All of those are extensively regulated by hormones. So glycogen for now, what we need to know, major glucose storage reservoir in the liver. Liver metabolism and glycogen go hand in hand. Lots of alpha 1,6 branching, and also obviously an alpha 1,4 glycosidic bond that joins them together. There's a whole host of diseases known as glycogen storage diseases, and that's something and maybe you'll learn in your medical school or a higher level biochemistry class. My advanced biochemistry, we go in depth into glycogen metabolism. For now, what we need to know about glycogen, how important it is for the body, is that this breakdown or incorporation of glucose into the glycogen storage form is regulated. Two of the most important hormones in that regulation are insulin. Glucagon, sort of the fight or flight hormone, actually stimulates liberation of free glucose. Insulin, on the other hand, stimulates the incorporation or the sequestration of glucose into glycogen. Glucagon and insulin are what are known as reciprocally regulated. They counteract one another. So the insulin hormone secreted by the pancreas will do one thing. To sort of offset that, the pancreas cells will, hey, slow down, revert back. That is when the hormone glucagon is released. So very fine-tuned according to the physiology of the body. This is something we do not need to know in Biochemistry 1, how insulin and glucagon regulate this. What we need to know about glycogen is the alpha 1,4 glycosidic polymer, alpha 1,4 bonds, the helical coil, and extensive alpha 1,6 branching, sort of making this sort of like a ball of yarn inside the liver cell. All right, here are some discriminating characteristics between amylopectin and glycogen. The main point here is the number of glucose monomers and the extent of branch points. So branching is more, it's happening at a greater frequency in glycogen. Plus, we also have a whole suite of enzymes that break down or synthesize glycogen. So this is far more complicated glycogen than amylopectin, but both have the alpha 1,4 linkage, and they both have alpha 1,6 branching. Let's talk about other types of polysaccharides. Here, let's focus on things that are also polymers, also polysaccharides that have different aspects in your physiology. As an example, we're going to show one polymer that's really not important in human physiology, but it's very important in exoskeleton of various types of insects and invertebrates, and this is chitin. So chitin is a very tough molecule. Point being here is that beta-1,4 glycosidic bonds. The point of this lecture is to show you the vast array of polysaccharides, and I want you to be very clear here that we describe them via a glycosidic bond where we specifically designate whether the beta anomer or the alpha anomer is making the bond. So here we have an beta-1,4. Again, this is a beta anomer of N-Acetyl-Glucosamine forming that bond with carbon number 4. Carbon number 4 is not anything. It's not anomeric. So it just is 4. N-Acetyl-Glucosamine, this is the N-acetyl group. For those of you that are in organic chemistry, the C double bond OCH3 is acetyl. Attached to the nitrogen is N-acetyl. And N-Acetyl-Glucosamine. This used to be the acetyl group is attached to the nitrogen. So a polymer of this, very tough, very rigid, kind of like your cellulose, except we have a derivatized form of glucose, and we call this chitin. Polymer of N-Acetyl-Glucosamine. Okay, the D-stereochemistry, just as a reminder, is determined by this carbon number 5, the chiral carbon farthest away from the anomeric carbon, and that substituent, CH2OH, is pointing up. Because it's pointing up, it's the D form. If it was pointing down, it would be the L form. Now here are some polymers that are very important in our body, in mammalian physiology, more specifically the extracellular matrix. These are glycosaminoglycans. So when you think of glycosaminoglycans, glycans kind of means sugar. And amino, a lot of these sugars are going to have amino groups. And these glycosaminoglycans are characterized by having a very much negative charge. So if you see a negative charge, nitrogen, sulfur, we see a lot of there, a lot of polar groups attached to these highly derivatized sugars, then we're thinking glycosaminoglycans. So associate this with negative charge, also associate this with the extracellular matrix. So what is the extracellular matrix? Kind of like a sponge. Okay, so things outside of your cell, things outside of your bone need to be very flexible. They must be like a sponge. They must take in water or squeeze out water. So that sponginess that allows us to jump or squat or twirl or do whatever it is we're doing it requires glycosaminoglycans. Also requires a whole host of other molecules that encompass the extracellular matrix. Collagen is also involved. Calcium is also involved. But as one part of this viscous gel, glycosaminoglycans are very important. So viscosity, mucus-like material, sponginess, flexibility, elasticity. These sugars, polymers of these sugars, very negatively charged, allow that to happen. So associate glycosaminoglycans with a negatively charged. Once again, we're going to see a lot of sulfurs, nitrogens in these polymers. Very important in mammalian physiology. So sponginess, flexibility, viscosity, hydration, thick, mucusy types of things. That's what the extracellular matrix is all about. We're going to talk about glycosaminoglycans here. Our first one here is hyaluronic acid. That's our first example. Okay, 250 to 250,000 polymeric units of B-glucouronate. Okay, so this is glucose, except we have a carboxylate here with a beta-1,3. Okay, notice that beta-1,3. This is the beta form of glucouronate forming a glycosidic bond with the number 3 carbon of N-Acetyl-D-Glucosamine. So why is this D? It's D because the substituent on carbon number 5 is pointing up. And why is this 3 just a number 3? Okay, because that number 3 is not the anomeric carbon. It's the carbon number 1 that determines whether it's beta or alpha. Hyaluronic acid or hyaluronate is a beta-1,3 glycosidic bond. Functioning, again, associate this with viscosity, flexibility. These things associate with collagen, sort of line up your bone, line up blood vessel walls, allow elasticity and viscosity. Okay. areas outside of their cell, for them to be fully functioning, need to have a gel-like or mucus-like consistency. Okay, and hyaluronic acid helps in that. Another example of glycosaminoglycans is chondroitin 4-sulfate. So let's take a look at this. Again, want to emphasize in the blue box the negative charges of these species. Okay, so here we have a sulfur, negative charge, a carboxylate, negative charge, a lot of polarities with that acetyl group there, the oxygen specifically. So this is a beta-1,3 glycosidic bond again. So the beta form of D-glucuronate forming a glycosidic bond with carbon number 3 of this species, N-Acetyl-D-Glucosamine 4-sulfate. Okay, so this is the galactose sugar, an epimer of glucose, if you remember. And a polymer of this, thousands to hundreds of thousands of units, makes up chondroitin 4-sulfate. There's also something called chondroitin 6-sulfate. Again, same function as hyaluronic acid in the previous slide, viscosity, allowing for shearing force, allowing for flexion, kind of like a sponge. Our third example is something that's not related to the extracellular matrix, and that is heparin. So heparin is actually found mostly as a something that prevents blood from clotting. So if you ever draw blood or had your blood drawn, there's very special types of blood collecting tubes that have the bottom coated with heparin, so that when they take the blood and it's collected, the blood doesn't immediately clot. So a lot of tubes have heparin coated in them. So this in our body is not associated with the extracellular matrix. So this is more of something associated with hematology. But it is a glycosaminoglycan. It is L-iduronate 2-sulfate. So want to bring your attention to the L. Why is this L as opposed to D? In the previous examples, we saw a lot of D sugars. Okay, look at that. Okay, so it's pointing down on carbon number 5, so that makes it L. Alpha, okay, not beta, so this is carbon number 1. The anomeric carbon is in an alpha form, alpha 1,4 glycosidic bond, with this beast of a sugar, N-Acetyl-D-Glucosamine 6-sulfate. Okay, that's sulfurs, that negative charge. There's another negative charge with the carboxylate. Classic hallmark of a glycosaminoglycan. Again, function of heparin, anti-clotting factor, secreted by some mast cells, and not part of the extracellular matrix. All right, alpha 1,4 glycosidic bond. L stereochemistry on this part. This is a disaccharide polymer. Just like the other previous two glycosaminoglycans were. A lot of these are found in stores. A lot of these are found in nutritional, supplemental, vitamin stores. As far as their efficacy, who knows? But again, in terms of sugar, some are beta 1,4 bonds, some are alpha 1,3 bonds, some are beta 1,3 bonds. But these glycosaminoglycans, in association with proteoglycans, in association with collagen, in association with a whole host of other factors, make our extracellular matrix more fluid-like, more viscous-like, more like a sponge.